Before we start, let me define lipids. Lipid is an organic compound found in living organisms that is insoluble in water but soluble in non-polar organic solvent. Unlike other biomolecules, lipids do not have a common structural feature compounds that serve as a basis for defining such compounds. Let me also define lipid metabolism. Lipid metabolism is the synthesis and degradation of lipids in cells, involving the breakdown or storage of fat for energy. These fats are obtained from consuming food and absorbing them, or they are synthesized by animals' liver. Today, we will be talking about the digestion and absorption of lipids, thoracic glycerol storage and mobilization, and lastly, the glycerol metabolism. The digestion and absorption of lipids. Digestion of lipids begins in the mouth, stomach, and small intestine. And absorption of digested lipids are absorbed into small intestine and then transport throughout the body. The body uses the digested lipids for many functions, including building cell membranes, making myelin sheets, insulating the body, storing energy, absorbing vitamins, making hormones. Thoracic glycerol are digested or hydrolyzed in the intestine and then reassembled after passage into intestinal wall. Chiromic runs transport the reassembled thoracic glycerol from intestinal cell to the bloodstream. Lipids play an extremely important role in cellular metabolism because they represent an energy-rich fuel that can be stored in large amount in adipose or fat tissue.
Triglycerol are a primary energy source stored in the body, and their mobilization and storage are tightly regulated to maintain energy homeostasis. Tag storage example: postprandial state, insulin stimulation, and adipocyte differentiation. Tag mobilization example: fasting states, exercise, and cold exposure. Triglycerol stored in the adipose tissue can be rapidly mobilized by the hydrolytic action of the adipocyte. The non-esterified fatty acid release are used by other tissue during time of energy deprivation. Triglycerides are a common type of a fat that account for about 95% of all dietary fats. Both animals and vegetable fats contain triglycerides circulate in the bloodstream to be used as energy by the cells. Any leftover are stored in the body fat to fuel the body between meals. Within all cell types, even those of the brain, triglycerol are stored as cytoplasmic liquid droplets enclosed by a monolayer phospholipid and hydrophobic protein, such as perilipins in adipose tissue or oleosins in seeds. Glycerol metabolism is the process through which glycerol, released from fat breakdown, is converted into energy and other compounds in the body. This occurs mainly in the liver and kidneys, where glycerol can be transformed into glucose or used in various metabolic pathways.
The main function of glycerol metabolism is energy source, lipid metabolism, and gluconeogenesis. Energy source is glycerol converter into glucose and used for energy production. Lipid metabolism is when glycogen precursor for triglyceride synthesis, which is stored in adipose tissue. Lastly, the gluconeogenesis, glycerol is converted into glucose, helping maintain blood sugar levels.
Glycerol metabolism. This is the metabolic pathway by which glycerol is catabolized. The glycerol hydrolyzed from fats can provide energy to cells. Glycerol is converted in the cytoplasm of the cell to dehydroxyacetone phosphate in 2 steps. Dehydroxyacetone phosphate is one of the chemical intermediates of glycolysis. Thus, glycerol, by entering glycolysis, can be converted to pyruvate and contribute to cellular energy production. The pyruvate formed from glycerol can also be converted to glucose through gluconeogenesis. So, during triglycerol mobilization, one molecule of glycerol is produced for each triglycerol completely hydrolyzed. Glycerol metabolism primarily involves processes considered in the previous chapter. After entering the bloodstream, glycerol travels to the liver or kidneys, where it is converted in a two-step process to dehydroxyacetone phosphate. First step involves phosphorylation of a primary hydroxyl group of the glycerol. In the second step, glycerol's secondary alcohol group C-2 is oxidized to a ketone. The following equation represents the overall reaction for the metabolism of glycerol. Glycerol plus ATP plus NAD plus dehydroxyacetone phosphate plus ADP plus NADH plus H plus dehydroxyacetone phosphate is an intermediate in both glycolysis and glyconeogenesis. It can be converted to pyruvate, then acetyl, CoA, and finally carbon dioxide, or it can be used to form glucose. Dehydroxyacetone phosphate formation from glycerol represents the first of several situations we will consider wherein carbohydrate and lipid metabolism are connected.